the NODCC is really excited to have sponsored this fellowship, and Ella's presenting over the next few months. Just to let everybody know, we are recording this and we will put it on our YouTube video. And then if you have any questions throughout the presentation, please just put it in the chat box and Ella will answer at the end of the presentation. So, Ella, I'll turn it over to you.
Sorry about that. Could not get the mute button to work or to work. Yeah. Thank you so much for being here today. Appreciate you all taking the time tonight. This is the second webinar in the webinar series in partnership with the NODCC. And the topic today will be pregnancy, birth, and medical issues in DCC. And to start off, I'm going to have Lynn say a few things, and then I will get into it.
Hi, everybody. Just to give a little context, once again, I'm Lynn Paul, for those of you who don't know me, and I have been around this world forever. And my research program is currently called the DCC Lifespan Studies, and we are looking at behavioral issues across the lifespan. Currently, our studies are focused from infancy through childhood, and that is the data that is informing the work that Ella is doing. Ella, if you want to go on. So these are parent reports provided through three approaches. One is we use standardized surveys and tests that are commonly used, validated measures. We also use a very commonly used standardized parent interview for the infant study. And then lastly, we're doing gather what we call natural history, which are background surveys that we developed specific to our study and to studies of the corpus callosum dysgenesis population. And we have developed these questionnaires in collaboration with other researchers so that we can share this background information. Ella. These studies are collected at whatever age the family enrolls, from 0 to 18 years old. And then we follow the kids every 6 months up to age 3, and then every year up to age 18. And the purpose of a natural history is to get a picture of what the experience is of someone with a particular diagnosis. In this case, it's disorders of the corpus callosum or dysgenesis. And to be able to objectively, using standardized approaches, find out what is typical of the symptom profile that kids and people have, what kind of diagnosis experience they have, and then ultimately, what are the interventions? So school, therapies, etc. So this is a study where we're not doing anything to intervene. We're not changing the experience. We're describing the experience. And the value of a natural history study is that it gives us a good picture of the variability in a group, the differences between subgroups, different types of diagnoses. And in our case, we're also looking at changes over time. So once we have these descriptions of the variations between individuals with the diagnosis and subgroups, we can start to identify patterns, and from those patterns come up with research studies, additional research studies to really test and understand the mechanisms that are involved in the challenges that these kiddos have, as well as the potential for different interventions. So that's just the context I wanted to provide at the outset, what a natural history is. all of you who have gone to a physician and gotten this diagnosis and said, what can I expect? What's going to happen? And they said, I have no idea. This is what we're trying to fix. We're trying to give objective data that physicians and clinical professionals will trust and will take all of your stories and turn it into data that they will then be able to tell a family, oh, we know that kids with this condition, blah, 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 blah. This is what to expect at 1, at 2, at 3, at 10. So, Ellie, you can move on. This webinar series is covering Ella's natural history, and I think I'm just going to hand it over to you from here.
Awesome. Thank you so much for that introduction. And to jump off of that, I'm going to give a little bit of an overview of the Natural History Project and what will be coming out of this. So this first sort of deliverable of this fellowship will be a series of webinars of this, which of this is the second one. 2 months ago, we had the first webinar, which talked about the power of patient experience and really did an overview of what natural history is about, as well as looked at some demographic information about the participants, as well as some diagnosis information. This webinar today will be covering pregnancy, birth, and medical issues. And then in the coming months, we'll have 2 additional webinars, one talking about the early years, so intervention and treatment and therapies and things like that, and developmental milestones, sorry. And the last webinar will be talking about later childhood, so emergence of autism spectrum disorder and going into school age and talking about everything related to that. So that's the webinar series that I'm presenting today, or the second of which I'm presenting today. And if you weren't able to be at the last webinar, there will be a link to it at the end of this presentation, but I will give a synopsis so that we're all caught up to go into this one together. So as I mentioned before, the first webinar was talking generally about the aim of the natural history study. And the aim is to define really what it means to have a disorder of the corpus callosum during development. And our main takeaway from this last webinar is that participation of anybody who is able to or wants to participate is ideal. Disorders of the corpus callosum or dysgenesis of the corpus callosum is a very wide diagnosis, and there are a lot of varied experiences, and we want to hear all about those, about all kinds of experiences, all ability and developmental levels, for as many ages as we're able to hear about, and all details and records. So yeah, that is a little bit about what we talked about in the previous webinar. I also mentioned that we talked a little bit about our participants in the last webinar, so I'll do a little catch up of what our participant sample looks like. So overall, we have 490 participants who have completed the full background questionnaire that Lynn mentioned before, which is awesome. So many participants. And of those 490 participants, 311 of them have complete ACC, 67 or 13% have partial ACC, 10% have hypoplasia, and 13% have dysgenesis. And here we also have our groupings, which I'll talk a little bit more about later in the webinar, but we have 2 groupings, which are ACC or DCC isolated, which means you have the disorder of the corpus callosum and maybe some commonly associated malformations of the brain, such as colpocephaly, an interhemispheric cyst, absence of the septum pellucidum, things like that, things that are commonly associated with a DCC. So that is isolated DCC. And we also have a second group, which is DCC plus. So these are participants who have a DCC as well as what we call a more atypical neuropathology. So these are things like heterotopia or microcephaly, abnormalities of the cerebellum or brainstem. So any diagnosis that really can have an impact on development, and a lot of the things that we're looking at in the study. So I'll be going forward and talking about DCC, isolated DCC and DCC plus. So that is that terminology. A big part of what we talked about in the last webinar as well is documentation in our study. So to be a part of the study, we ask that participants submit medical records, which are really important for publishing, as many journals require documentation of diagnosis. So it's important for publishing, but it's also important for us and to have quality of information in our study, so that we know what we are reporting. So there are sort of three scales here that we have, and participants sometimes don't submit any medical records, and this is difficult for us because we're unable to confirm that diagnosis. And again, we talked about publishing. It provides a barrier to being included in additional samples for publishing. Participants are also able to submit a scan report, which is awesome. We have that documentation, and it's from a primary medical source. But the issue is that we aren't able to see the scan ourselves, and by ourselves, I mean with our amazing team that reviews our medical records at UCSF. So we are unable to see the quality of the scan, and so while having a report is awesome, great documentation, what we really love to have is the brain scan itself. And again, I mentioned we have a team at UCSF, the University of California in San Francisco, working to review those scans. And it's really important that we have those scans reviewed by this team because they know disorders of the corpus callosum very well and are experts in the field, and so they can provide a really quality diagnosis to use for research, as well as very consistent. It's, all of the reviews are done by the same person, so this is sort of what we would really love to have is documentation of brain scans from everybody. But yes, so this brings us to our next point, which is our sample that has a verified diagnosis. So what we consider a verified diagnosis is participants that submit a postnatal MRI report. So anytime after birth, they have either an MRI scan, and they submit at least the report and also the scan to us, if possible. And this brings our total participation down to 249 participants. So as you can see here, in this pie chart right here, about half of that sample is unable to be verified, and so it is more difficult to look into subgroups. So maybe if there's something being caused by an atypical malformation, as opposed to more of a common malformation, we aren't able to have as large of a participant sample to look into, to be able to parse through that, as well as it's harder to look through difference in diagnosis. So we have smaller numbers of each type of DCC diagnosis. So overall, we would really love to get those numbers up in terms of verifying our diagnoses of our participants. But I wanted to introduce those two samples to you because we'll be using both throughout this presentation. And this will be indicated by one of these two circles right here. So if you see a yellow circle in the top right corner, this will be our full sample. So everybody who has that full background. And if you see the blue circle in the right-hand corner, that will be our verified sample. And our reasoning for using these in different situations is some of this information we aren't looking at through the lens of diagnosis. So coming up, you'll see pregnancy history and birth history. We aren't looking into subgroups for that, so we'll be using the full sample, whereas things like other issues we'll be looking at with the verified samples so that we can see subgroups of isolated and plus. So that is just a going to be coming up. All right. And I also want to make a note that in this presentation, these are preliminary findings. And so there will be a lot more information being analyzed throughout the next couple months. And this is just sort of a snapshot of what the information looks like at this moment. Additionally, in the previous webinar, we talked a little bit about consistency of diagnosis over time. So a lot of our participants were diagnosed prenatally. So they had a scan in utero, and then they are often have additional scans postnatally, but we don't always have access to those. And so we were looking at a really important question, which is, what is the consistency of diagnosis over time? And this is important because, again, since so many people are diagnosed prenatally, sometimes they only have that scan, and we want to know how accurate that diagnosis itself is. However, due to limitations in our current sample, due to multiple things, we are unable to find a more concrete answer at the moment, but it will be a subject of future study. So our first finding that I want to talk about today are our genetic findings. And so, as it says in the right-hand corner, this is our full sample as we are not parsing out any subgroups. So out of our 490 participants, 79% of them have had genetic testing. And of that 79%, or 388 participants, 20% of them have had a confirmed genetic finding. And of those 80 participants, they have a genetic disorder related to DCC. So this can be something causal or something that, or a finding that has association with DCC. Okay. And along with genetics, I wanted to talk about our families within our study. So currently in our 490 participants, we have 14 who are twins. And so on the left, we have our identical twins, and on the right, we have our fraternal twins. And not all of our sets of twins both have DCC, but some of them do. And so as you can see here, with each pairing, I have listed the diagnosis of the individual twin. So this first one up here, one twin has partial agenesis and the other twin has hypoplasia. And then the one below that, the one twin has dysgenesis and one has is a sibling who does not have dysgenesis. And these sets of twins and siblings going forward provide a really awesome base of information to look at differences between individuals with all of the same environmental and genetic backgrounds. So as I mentioned before, we have some sibling pairs as well. So 10 out of our 490 participants are related to other individuals in the study. So that has we have 5 sets of siblings, and all of the sets of siblings do both have a disorder of the corpus callosum. Additionally, in our study, we have multiple people who have reported other relatives that are not a part of our study, but do also have a relative with a disorder of the corpus callosum. So 4 individuals reported that they have a cousin with a disorder of the corpus callosum. 3 indicated that they have a sister with DCC, and 1 indicated that they have a grandmother with DCC. And these familial findings are all a really great base of beginning to understand genetic relations, and it is a good base for future study. And I'm going to get into pregnancy and birth history. And this again will be with our full participant sample because we will not be looking into any subgroups. sorry, we will be looking into subgroups. Within the pregnancy complications, more often, more isolated participants experienced these pregnancy complications on average, which makes sense because we do have more isolated participants than DCC plus participants. Are also looking at delivery and perinatal complications. So things like jaundice, feeding issues, respiratory issues, congenital defects, infections, cardiac malformations, apnea, neurologic issues, cyanosis, stroke, and seizures. And yeah. We also are looking into newborn measurements. So these, in the blue circle on the left, we have our sample average. And then on the right, in the pink circle, we have our published normed average. And by R, I mean a published norm average. And our participants compare within pretty much the range of all of these measurements. And so this is for females, assigned female at birth, and these are for assigned male at birth, just a little bit different. And as we also talked about in the previous webinar, sometimes statistics can be deceiving. So if an average is looking a little low, we might want to look into why that is happening. And so in this case, we wanted to highlight that birth weight can have a lot of reasons for being maybe lower or higher. And one reason is that gestation varies across, or gestational age varies across our participant pool. And so if a participant is born at a lower gestational age, they will likely have a lower birth weight. And this is confirmed, as you can see in this figure. And a similar finding with birth weight and head circumference. So a lower birth weight may indicate a lower head circumference. And next, I'm going to talk a little bit about sensory systems. So for our participant pool, we are including the full participant pool in this discussion of hearing because we looked into subgroups and there were little to no differences, and so we decided to use the larger sample. So out of our 490 participants, 93% of them did have a hearing test, and of those participants with a hearing test, 86% of the hearing tests were normal or had normal results, and 13% of them had abnormal results. And of those 13, or 12.5% of participants who had an abnormal hearing test, they largely were not sure what kind of hearing loss their child was experiencing. But those who did know, 21% were sensorineural, sorry, that was hard for me. And 8% had conductive hearing loss, and 1% had both sensorineural and conductive hearing loss. So next we're going to be talking a little bit about vision. And for this, for vision, we will be using the smaller sample because we will be parsing out those subgroups of isolated DCC and DCC plus. So with this figure, I know there's a lot of information happening, but I'll break it down. So as you can see in each part of the Venn diagram, there are two numbers, and on the left, we have our isolated DCC percentage, and on the right, we have, in parentheses, we have the DCC plus number. And so with visual abnormalities of individuals with optic nerve hypoplasia, which I will talk a little more about in the next slide, there was a lot of overlap with other visual abnormalities, so we decided to look at the overlap between all three. And yeah, they're very interesting findings, and again, these will be more continued to parse through in the coming months. Optic nerve hypoplasia is, I wanted to touch a little bit more on this because there is an association with DCC. Optic nerve hypoplasia or optic nerve malformations are a congenital malformation that results in an underdeveloped optic nerve, and this commonly co-occurs with DCC and can cause visual impairment. This is also a hallmark of another disorder known as septo-optic dysplasia, which is also associated with DCC, as septo-optic dysplasia consists of malformations of at least two of the following: the optic nerve, midline brain structures, including the corpus callosum, and the pituitary gland. And within our participant sample, our smaller participant sample, so that 294, 249, there we go. 11.6% of our participants did report a finding of optic nerve hypoplasia, with that being a little bit higher in our DCC plus participants, so 3.5% in isolated DCC participants and 8% in DCC plus participants. And I also wanted to talk about something that will be coming up in the next webinar, but I wanted to give a little preview about it now. Today we're talking mostly about physical and neurological issues and abnormalities within our participants, but these are important to note because they have a lot of effects on other experiences of our participants. So an individual experiencing visual issues may have issues with depth perception, visual tracking, and acuity, whereas somebody with hearing loss may have difficulties with sound localization, and individuals with deficits in other senses may have difficulties with pain perception and temperature regulation. And these are all important to know because they can affect living quality, and it's good to know so that interventions are able to be taken. So I will next be talking about other biological systems and diagnoses. One thing I wanted to touch on again in a preview of the next webinar will be muscle tone. And this will be, again, coming from our larger sample of 490 participants, but 3.7% of them reported high tone only, and 34% reported only low tone, and 21% reported having both low and high muscle tone in different parts of the body, and about 50% of our participants reported no tone-related issues. But I bring up tone because it can have a lot of impact on things like physical milestones, walking and sitting, as well as more fine motor skills. And so it'll be really important in our next webinar, and I wanted to touch on it because it is a physical abnormality or a physical issue that can have complications for developmental purposes. I also wanted to report that about 16% of our participants reported cardiac issues, and 19% reported respiratory issues as well. And so our last section will be talking about neurological conditions and seizures. And so this will be, again, with our smaller sample, our verified sample, as we'll be looking at differences between isolated and plus participants. For neurological complications, this first one on the left down here that you can't see is insensitivity to pain, which we talked a little bit about before, as well as involuntary movements that also got cut off, and temperature dysregulation, as well as headaches, tremor, tics, dizziness, and numbness. And in this figure, we can see that the participants with DCC plus are reporting higher incidences of these neurological issues. And this can be for multitude of reasons, including it can be related to other atypical malformations. And so next, before I talk a little bit about our rate of seizures in our participants, I wanted to talk about the different types of seizures that we will be discussing in the following slides. And I thought I would break it down in terms of what the topic or what the terms are related to. And so over here we have locations. So you might hear the term focal seizures, and this is related to seizures that have activity restricted to one portion of the brain, whereas generalized seizures are when activity inhabits both hemispheres of the brain. You also hear febrile and afebrile. So febrile is a seizure that's caused by fever or illness, whereas afebrile is not caused by fever or illness. We will also be talking about infantile spasms, and these are seizures that are specific to babies under 12 months, 12 months or under. And we'll also be talking about absence seizures, which are the name is related to its symptomology, meaning there is a lapse in consciousness. And then there's atonic, which is a loss of muscle tone, and then tonic, which is a stiffening of the muscles. And so I just wanted to give a little overview before I get into the next couple of slides. So within our participant pool, and this is the larger participant pool, 16% of our participants have experienced at least one seizure in their lifetime. And of those 16% or 82 participants, 75 of them have experienced multiple seizures. And of those 75% or 62 participants, 75% of those participants have had multiple seizures in a month. So at their most active seizure state, they experienced at least 2 seizures in 1 month. Of our 16% of participants that have experienced at least 1 seizure, this figure is depicting the age of their first seizure or the onset age of their seizures. A large majority of our participant pool experiences their onset of seizures before 2 years, with particular emphasis between 6 months and 24 months, which seems to be a point when a lot of our participants are experiencing the onset of their first seizure. And again, going back to these seizure types, of our 16% of participants that experienced at least 1 seizure, 28 of them experienced generalized seizures, whereas 13 of them experienced focal seizures. 19 people experienced febrile seizures, 18 experienced absence seizures, 14 experienced atonic seizures or infantile spasms, 11 participants experienced other types of seizures, and then 17 participants experienced seizures but aren't sure what type they are. I also wanted to talk a little bit about seizure prevention tools, because there are well-documented triggers of seizures, and so it's important to be prepared and have these things in your toolkit to know what could be affecting your child or yourself in the onset of these seizures. And so some things that are documented to cause seizures or trigger seizures are dehydration or lack of nourishment, stress, mental or physical stress, lack of sleep, flashing lights, and substance use. So these are all important things to keep in mind for individuals with a disorder of the corpus callosum, as there is a higher incidence rate of seizures within this population than on average. In terms of interventions for seizures, it's important to seek out professional interventions, like neurologists, and utilize EEG evaluations in multiple states, as seizures are able to occur in any state, being it sleep or awake, and during stress. So having these tests to know your particular self is important. And I wanted to talk again a little bit about future variables and how everything we talked about today connects to things going forward. So we'll be continuing a little bit of medical history in terms of psychological health, and future webinars talking about ASD diagnoses, other neurodevelopmental diagnoses, as well as psychiatric diagnoses. And we'll also be talking about developmental history, so age of first words, steps, social skills, school age, or school skills and interactions in school, as well as issues with sleep and issues with feeding. And a lot of things we talked about today will contribute to that, such as we talked about tone. This can have an effect on feeding in terms of breastfeeding. Tone can have an effect on developmental milestones. And there are a whole bunch of things that, again, we talked about today that will contribute to the larger story of the natural history study and what DCC looks like in development. And today we were doing a lot of establishing sort of what participants' physical experiences have been, and we'll continue to build off of that in future webinars. And so some takeaways are today we continued to establish a baseline for our participants' experiences. Most notably, 16% of our sample, our participants have experienced a seizure. 16% have also reported a significant genetic finding, and 41% have reported visual abnormalities. And another takeaway is the information covered today is just the beginning of the story, as I've mentioned a couple of times. There's so much to draw on in these physical characteristics and how they contribute to living life and many, many things. So this is, again, just the beginning, and there's a lot more to talk about in the future. And I've mentioned it at the beginning, but the upcoming webinars, there will be one in, those dates are wrong, sorry about that. There will be one in May talking about supporting development and intervention, and then there will be one in June around the same time, the middle of the month, talking about the emergence of ASD and other diagnoses in later childhood. And thank you to everyone who's a part of the lifespan studies. Thank you, Lynn, for all of your help. And thank you so much to the DCC for continuing to support this fellowship and continuing to support this work, this very valuable work coming to light. And thank you for everyone who has participated in this research program. Again, we could not do it without your many, many hours of survey answering and phone calls. So I very much appreciate your help, and yes, thank you. And if you're not already enrolled in our study, we would love to have you. So on the left, we have a QR code to enroll now. And if you're not sure if you're already in our study, I know there are a lot of studies around. So you can scan the QR code on the right and send us an email, and we'll let you know if you're already enrolled.
Thank you, Ella. I want to commend Ella. She has really jumped into this wholeheartedly at a much deeper level over the last month. And this is a huge data set. And it's easy to say we want to ask all these questions and gather all the information, but then to synthesize it and discern how to make the most sense of it is a really complicated process. And she has put in a lot of extra time trying to do that for this presentation and going forward. So I'm excited to see what's coming out of it and where this will take the entire field and be able to inform families. I'm curious if any of you have questions for us at this point. Were there any things that surprised you, Ella? Ella's done a lot of the parent interviews, so she's talked to a lot of families.
I'm trying to think of anything that comes to mind. I think I know I keep talking about tone, but muscle tone often comes up in survey answers and in phone calls and phone interviews. And so I think seeing those numbers and seeing that there is a high incidence of loss of muscle tone, I think it felt validating to a lot of people's experiences, that is a shared experience.
Yeah, that was one that stuck out to me. I was a little surprised that the visual impairment wasn't higher,
Yeah.
but one of the things that you may have noticed as you're listening is there are places where you'd like to have a better idea of what's normal, what in a non-neurological population is standard and incidence. So there were a few of those numbers that we don't have yet, and that will definitely shape how we present the data in the online tool and in the research papers. But yet you can also see from Ella's comparison of something like uh, brains, I think it was the birth statistics, that they're at least at the average, they're not looking dramatically different than average. But one of the things that we're going to look at down the line is what's the spread? What's the variation? And most likely, our population is going to be a lot more variable in these things than a typical population. And so those are the kind of questions that we will be continuing to follow up on.
I wonder if the visual is based on if they're getting recommendations to get the vision checked, number one, as well as at what age they're getting it done. That's another question too. So you're muted.
Sorry, there's a clock in the background. Um, I think, Ella, you were including the entire sample. And for vision, we really do need to be aware of the age because they're not going to have acuity testing until there's enough acuity to test. So
Yes, but
it,
that's important information to present to parents, especially those that are just getting started in their journey to, you know, making that appointment to check is important.
Hey, this is Kim Manning. Possibly because my son was premature, but we had him to a pediatric ophthalmologist probably when he was just a couple months old. There aren't many of them, but it was to see if there was retinopathy of prematurity, to see if that had, here's my clock for you, to see if that had impacted him. And really, well, they never did the, is it A or B, which you see better on? They did do a lot of testing on him um, at a very young age. Just throw that in.
Also, if they're looking for it in the MRI, because we were diagnosed via an MRI, so for the optic nerve.
You're muted.
sorry. It points out another issue in doing natural history, which is um, when you start talking about systems, such as vision, there are tests that are structural tests and there's tests that are functional. And Ella was talking a lot about um, things that are structural that then are going to have functional consequences, or things that are physical that are going to have more functional activities of daily living consequences, even though it's a limited scope. Um, but we have to be careful when we talk about vision impairment or vision issues to be clear about, are we talking about visual anatomy and a disruption in the physical system and or the functional systems?
This is Joe. A lot of great data and information. Ella, I just wonder, sort of if there's almost a best practices list of potential um, issues to test for, or at least think about testing for parents who have just been given the diagnosis. And I know it's not a sort of 0-1 kind of thing, but I imagine some of this data would, statistically anyway, start pointing to certain tests being good to think about doing along the journey. I think, Barb and Kim have mentioned different things that have been done, and I'm in the same boat as a parent, but it would be fantastic to be able to provide almost like a potential checklist. And obviously, no one checklist is going to be comprehensive for everyone or applicable to everyone, but I think some of us have had to figure out the checklist along the journey.
I think that's a great point, Jyotish, because that's definitely something that we want to be a conclusion of this natural history is start to point to these are high incidence issues that need to be considered when the diagnosis happens. Yeah, thank you for bringing that up. Of which cardiac, I mean, the issue is midline, and so we know that these midline conditions are often associated, but I was surprised at how high the incidence was.
There are a couple specific cardiac malformations that I've noticed. Um, I can't think of any off the top of my head, of course, but going forward, I will look at that. And yeah, I think it was a higher incidence rate than I expected there to be.
Yeah, that's really interesting. In 25 years of doing this, I've only heard of a few cardiac kiddos, so
I did have one patient whose heart was flipped. The left and right were flipped.
hmm.
Yeah. The other thing I thought was kind of fascinating was the number of the twins, that all of the fraternal twins were concordant for having a DCC, but identical were not. Um, which really points to the de novo mutation. So a mutation that happens in one child, one fetus, after that is not linked to the DNA that they are carry with them originally, but is a mutation that happens once they're formed. There's a question. We don't have any data about astigmatism surgeries. We asked about astigmatism or strabismus surgery. I don't know about astigmatism surgery. I don't really know anything about that.
We did not ask particularly about.
Oh, so
I,
you didn't mean astigmatism is a waving of the top outside of the eye. You're talking about amblyopia or strabismus surgery. So when you've got a lazy eye, and there is a higher incidence of that condition, but I don't know about the surgeries. Ella?
um, we have some information from participants about that they had a surgery or that they were looking to avoid a surgery, but nothing um, specific that we asked in particular. But there were some reports of that.
That's a great example of the number of questions we could keep going with. 15-year-old with complete ACC has a twin without DCC. Are these identical or fraternal? Fraternal. So if it's fraternal, um, it could be any of a really broad range of things. It could be, like I was saying, a de novo mutation in one of the children, not the other. It could be something that is inherited. So it is something that one of the parents or both are carriers, and it just happens to manifest in one child and not the other. So that's, um, I think if it's a fraternal, then there are more variations of what it could be than if they're identical. And because you're not going to have identical twins with a passed down genetic condition or cause because they have the same DNA. So it had to have happened after that. So fraternal, it's just much more open-ended. We don't know. And in our sample thus far, the number or the rate of genetic findings is low. I will tell you that in a paper we're working on right now in a large collaboration across sites with Washington University and UCSF and my group, um, in our cohort of about 500, 600 cases that we have full genetic testing, the hit rate overall of identifying a causal gene is about 50%, which is really high for a condition. It is much lower in general within the isolated complete cases. It is much higher than 50% in hypoplasia. So we're starting to be able to see some patterns of where at least at this point, it looks like the genetic causes are going to be more informative. But there's probably a lot of kids with isolated or and particularly complete absence that it may be something that's it's not inherited genetics, and it may even be something that happens from an environmental effect or something. So many questions to answer. Um, One thing I will say about the gaze issue, since you brought up astigmatism, um, I mean, Barb, I can talk about the issue of depth perception. If your eyes are not aligned, you can, tell if something's in front of or behind by occlusion, if it hides, but you can't tell just by a sense of depth, stereo vision. And so something like walking downstairs can be difficult, noticing when the surfaces are uneven. So it puts a child at greater risk of falling. Socially, it also makes it really difficult. So often when we're interacting socially, we look at something and we want the other person to look at it with us. And even if a child gets the idea that your head is looking in that direction, we can glance at someone's eyes and have an idea of how far away they are looking. It's phenomenal when you start to pay attention to it. And these kids see the world in 2D. So you may say it's in front or behind, and to them, that means nothing. Because if it's 2D, you got to tell them it's above, below, right, or left. So they can have this experience of being directed either to share an experience or share attention or do something, and they have no idea what you're talking about because they don't have that sense. They don't have the sense of depth to even recognize the cue. Um, so just food for thought for those of you who deal with that.
Yes. It's a
And it's,
treat.
yeah, and it's really hard to start thinking in
Yeah.
terms of above, below, right, left when,
Yeah. Well, and they're processing not only the vision part, but the words in different ways too. So
yeah.
that's the other thing. We have a son who has incredible depth perception, so he does not look down when he walks, and it's the scariest thing you've ever seen. He doesn't go to look down for steps. He just, uh, I have no idea. So yeah, we
He
could go
does
for
have,
hours. He does not look down. Mm-mm. His depth perception is that crazy.
it's that good?
When we say look down, he will not look down. He just knows like where the step is.
Wow.
Yeah, it's insane. I'm like, please look down. But
Hmm.
but because he can't visualize sometimes like the height, that becomes a problem. Like he knows the step is there, but he doesn't know how deep the step or how shallow the step is too. So a lot of times we hold on to him as he's going down the stairs. He doesn't have to look down to see them, but we're grabbing
Well,
onto him because sometimes they're shorter or deeper or those
and
are the
that's
issues.
what I think, Barb, what you're getting at is that doesn't have any meaning to him. So
Mm-mm,
if you say look down, why?
mhm.
Why bother? It doesn't mean anything. It gives
Yep.
him no additional information.
But it's interesting because in doing escalators, he doesn't love to do escalators. And I think because he doesn't understand like
Same thing. He doesn't
when to
get the
step,
dimension of
when
it.
to get off. Yeah, he doesn't get the dimension of it. Yep.
Yeah.
So
So, yeah.
it's
Anyway,
scary,
we could
but
go on and
he
on.
does it. He functions, but
Well, thank you, Ella, for all
yes.
of your work on this. And thank you again, over and over again, to the DCC for supporting Ella and supporting this project. We hope to have a lot more to offer in the coming months.
We're looking forward to it. So thanks you guys for putting this together, and we will see you next month again.
All right.